Dear friends, good morning and welcome to a new episode of my video series on MIT Homeopathy. Today, I am trying to explain the biological mechanism of curative process involved in the fundamental homeopathic principle similia similibus curantur in terms of modern scientific concepts and paradigms. Hope all of you will agree this is highly essential and vital for establishing homeopathy as a scientific medical system in this modern era of enlightenment. By similia similibus curator, Samuel Hahnemann, the great founder of homeopathy, says that a substance can cure a disease if the symptoms produced by that substance in healthy individuals are similar to the symptoms expressed by the person in disease condition. Looking from a scientific point of view, similarity of symptoms indicate similarity of affected biomolecular pathways, similarity of molecular inhibitions, similarity of target molecules, similarity of involved drug molecules and pathogenic molecules, and ultimately similarity of their functional groups. In order to be capable of explaining similia similibus curantur scientifically, first of all, we have to study carefully the phenomenon known as competitive inhibitions in modern biochemistry. I would like to request all those scientific critics of homeopathy who are known as skeptics, who declare homeopathy is unscientific, homeopathy is mere placebo, homeopathy is implausible, to study this phenomenon once again, well explained in your biochemistry textbooks, and try to relate it with the concept of similia similibus curantur. If you do it truthfully, you will realize how much blind and prejudiced you were so far in declaring homeopathy is unscientific. As all of us know, competitive inhibition is the interruption of a biochemical pathway owing to one chemical substance inhibiting the effect of another by competing with it for binding or bonding with same targets due to the similarity of their functional groups. Any metabolic or chemical messenger system belonging to our life processes can potentially be affected by this phenomenon, but several classes of competitive inhibition are especially important in biochemistry and medicine, including the competitive form of enzyme inhibition, the competitive form of receptor antagonism, the competitive form of anti-metabolite activity, and the competitive form of poisoning, which can of course include any of the aforementioned types. In competitive inhibition of enzyme catalysis, binding of an inhibitor prevents binding of the natural target molecule of the enzyme, also known as the substrate. This is accomplished by blocking the binding site of enzyme, also known as the active site, where the natural ligands or substrates are expected to bind with. Competitive inhibition can be overcome by adding more substrates or natural ligands to the reaction, which increases the chance of enzyme and substrate binding. This is known as reversibility of competitive inhibitions. In competitive inhibition, An inhibitor having functional group similar to the normal substrate or ligand binds to the enzyme, usually at the active site, and prevents the substrate from binding. During competitive inhibition, the inhibitor and substrate compete for same active site on the enzyme. The active site is a region on an enzyme which a particular protein or substrate can bind to. The active site will only allow one of the two complexes to bind to the site, and therefore either allowing or a reaction to occur or yielding it. In a state of competitive inhibition, the inhibitor molecules resemble the substrate and therefore takes its place, thereby binding to the active site of an enzyme. Competitive inhibitors are commonly used to make pharmaceuticals. For example, methotrexate is a chemotherapy drug that acts as a competitive inhibitor and is used as a medicine for cancer treatment. It is structurally similar to the coenzyme called folate, which binds to the enzyme dihydrofolate reductase. This enzyme is part of the synthesis of DNA and RNA, and when methotrexate binds the enzyme, it renders it inactive so that it cannot synthesize DNA and RNA. Thus, the cancer cells are unable to grow and divide when methotrexate is used. This is how methotrexate acts as an anti-cancer drug. Another example of competitive inhibition involves prostaglandins, which are made in large amounts in our body as a response to pain and can cause inflammatory processes. Essential fatty acids form prostaglandins, and when this was discovered, it turned out that these essential fatty acids are actually very good inhibitors of prostaglandins. These fatty acids inhibitors have been used as drugs to relieve pain because they can mimic as the substrate and bind to the enzyme and block prostaglandins due to their similar functional groups. Actually, it is this phenomenon of competitive inhibitions that works behind similar concept of homeopathy. It actually means a molecular inhibition produced by a particular pathogenic molecule could be removed by utilizing a drug molecule having competitive relationship with it due to the similarity of functional groups. If the functional group of pathogenic molecules and drugs molecules are similar, they can bind to similar molecular targets and produce similar symptoms. That is why homeopathy tries to identify similarity between pathogenic molecules and drug molecules by observing the similarity of symptoms they produce in living organism. Through the principle of similia similibus curantur, Hahnemann was actually trying to explain and utilize this phenomenon of competitive inhibition for the purpose of developing his new therapeutic method. When we try to remove molecular inhibitions in a disease condition by using competitive inhibitors as done in allopathy, there is always a chance for developing new drug-induced diseases due to their off-target actions in the body. This phenomenon underlies the dangerous side effects of most of the chemotherapeutic drugs and allopathic drugs. It means when we use molecular forms of simillimum or competitive inhibitors for treating the disease, it may lead to establishing new diseases that may be more harmful to the organism. Hahnemann also observed this possibility of drug induced diseases, and he tried to overcome this danger by using potentized forms of competitive inhibitors or simillimum. In order to overcome these adverse effects of off-target actions of competitive inhibitors when used for therapeutic purpose, Samuel Hahnemann developed the technology of drug potentization. Homeopathic potentization involves the process of preparing molecular imprints of drug molecules in water ethyl alcohol matrix using drug molecules as templates for imprinting. Molecular imprints are supramolecular clusters formed in the imprinting medium, wherein the spatial conformations of template molecules remain engraved as nanocavities. Due to complementary conformations, these molecular imprints of competitive inhibitors can act as artificial binding sites or pockets for the pathogenic molecules and deactivate them, thereby removing the pathological molecular inhibitions they have produced in biological molecules. If symptoms produced in healthy persons by a drug substance taken in its molecular form are found to be similar to those expressed by an individual in a particular disease condition, that drug substance, if applied in molecular imprinted forms, can cure the particular disease condition of that individual. It is homeopathy. If symptoms produced in healthy individuals by a drug substance appear similar to the symptoms expressed in a disease condition, it obviously means that the particular drug substance as well as the particular disease causing molecules contain some chemical molecules having similar functional groups of or moieties, so that both of them were capable of binding to same biological targets in the organism, producing similar molecular errors that are expressed through similar trains of symptoms. Molecular imprints of similar chemical molecules can act as artificial binding pockets for similar chemical molecules and deactivate them due to their mutually complementary conformations. Due to the historical limitations of scientific knowledge available to him during his time, Hahnemann could not understand that two different substances produce similar symptoms when both substances contained chemical molecules having functional groups or moieties of similar conformations, by which they could bind to similar biological targets and produce similar molecular inhibitions that lead to similar biological errors in similar biological pathways. Remember, Hahnemann was working during a period when modern biochemistry has not even evolved. It is obvious why Hahnemann could not explain the phenomenon he observed using the paradigms of modern biochemistry. But his extraordinary genius could foresee its implications in therapeutics. When a homeopath searches for a simillimum for his patient by matching the disease symptoms and the drug symptoms, he is actually searching for a drug substance that contains some chemical molecules that are having conformations similar to those of the particular chemical molecule that caused the disease condition, so that the drug molecules and the disease causing molecules will have a competitive relationship in binding to the biological molecules inside the body. Since molecular imprints of drug substances contained in potentized forms of homeopathic drugs can act as artificial binding sites for the disease causing molecules having competitive relationships due to the conformational affinity between them, and remove the pathological molecular inhibitions. Post-Avogadro dilutions of simillimum drug could be used as a therapeutic agent as per the principle similia similibus curator, which is known as the fundamental principle of homeopathy. Homeopathy or similia similibus curator is a therapeutic approach based on identifying drug molecules that are conformationally similar and capable of competing with the disease causing molecules in binding to their biological targets by observing the similarity of disease symptoms as well as the symptoms drug substances could produce when applied on healthy individuals, and deactivating the disease causing molecules by binding to them using molecular imprints of similar drug molecules. I hope in this video I have explained the scientific basis of similia similibus curator in terms of competitive inhibitions, well explained in our biochemistry text. Thanks for listening. We'll come back with another video shortly. Goodbye.